Fantaмен özülüş jasa. Eksperiment jasada, özülüş boyu көңіл көтер. Шаттық формуласы Fanta плюс әлдену. Достарыңмен ойнаған сәттерің тарихта қалады. Özülüş математикасы оңай. Әлденіп ал, Fantaмен рахаттан. Қане, демалатын кез келді. Fantaмен әлденіп ал. Сабақ арасында Fantaмен әлденіп ал. Көңіл көтер, қайтала.
Олар жайлы фильм түсірілеміз жоқ па? Кітаптардағы немесе комикстердегі қаһармандар қатарынан табылама. Оның супер костюмын біреу армандай ма? Оның деген сөздері қанатты сөзге айналама. Оларға бұның ешқайсысы маңызды емес.
Мынау сізге.
Оларға әлемді сәлде болса жақсарту маңызды. Міне осындай адамдарға жусан керек. Жусан қосымша сен үшін, мүмкіндіктер сен үшін.
Сделай перерыв между занятиями с Фанта. Проведи эксперимент и развлекайся весь перерыв. Вот формула веселья. Фанта плюс перекусы. Ваши игры с друзьями войдут в историю. Математика перерыва проста. Получай удовольствие от Фанта и перекусов. Давай, уже пора передохнуть. Перекуси с Фанта. Перекуси с Фанта между занятиями. Повеселись, повтори.
Им не важно, снимут про них фильм или нет. Станут ли они героями книг или хотя бы комиксов? Мечтает ли кто-то про его суперкостюм? И с какой вероятностью его станет крылатой фразой? Им не важно.
Это вам.
Им важно делать мир немного лучше. Вот для кого нужен Жусан. Жусан. Приложение для тебя. Возможности для тебя.
S.A.S Kazakh Agritechnical University, the general partner of this event, is hosting this year's festival. For 65 years of its activity, KATU has trained more than 70,000 specialists, successfully working in all major sectors of the agrotechnical sphere. Among the ranks of our graduates are deputies of the Parliament of the Republic of Kazakhstan, heads of leading enterprises, nationally known public figures, and athletes. Today, our university is one of the leaders of the national higher education on the national rating of the demand for higher education institutions of Kazakhstan, from the IAAR on the quality of educational programs. And also, KATU is the base university for digitalization of agricultural production in Kazakhstan. Our partners include more than 160 world-renowned leading research universities and research centers. Among them are the University of California at Davis, which is the second highest institution in the world of the agricultural industry, Agroparitech University in Europe, and many others. Today, KATU has started a dual diploma education in 3 specialties jointly with the University of Milan in Italy, which gives our graduates the opportunity to receive diplomas from 2 countries. Our university offers the opportunity to study at bachelor's, master's, and doctorates. Currently, the number of students exceeds 12,000, of whom 70% are studying under the state grants. There is a full opportunity to study in the military department. All necessary conditions are created for them to be qualified specialists. The university aims for a seamless experience in any format of education. For students, there is a printing office and a library equipped with an electronic system that has more than 1.5 million book collections.
One of the large multidisciplinary education institutions of Kazakhstan with a deep history is S.A.S University. That is, here students and undergraduates acquire practical working skills. In other words, this is a mini copy of a large production and dairy plant. Within the framework of the program of scholarship academic mobility and double degree education, the university cooperates with more than 170 foreign universities. 150 students a year study for the academic mobility at leading universities in the world. The digitalization process has affected all spheres of society, including education. At S.A.S University, innovations are intensively introduced into the education system, and 3D, VR, STEM technologies are widely used. The IT Center provides consulting to teachers of schools in Shymkent on robotics, provides material resources to students in the Laboratory of Robotics and Mechatronics at the Laboratory of Mathematical and 3D Modeling, and provides material resources of the IT Center to students and teachers of the S.A.S University. In Student Design Bureau, students and teachers prepare scientific projects for participation in regional, national and international competitions. Students of clubs and Student Design Bureau develop robotic systems for participation in competitions and competitions in robotics. The material and technical base of the university consists of 10 educational buildings of a modern standard, 6 student dormitories and 2 sports complexes. For the comprehensive development of students, more than 30 public, political and cultural organizations function. S.A.S University, that has united history and modernity today, prepares more than 27,000 students and invites to take your best.
Сабақ арасында фантамен үзіліс жаса. Эксперимент жасады, үзіліс бойы көңіл көтер. Шаттық формуласы Фанта плюс әлдену. Достарыңмен ойнаған сәттерің тарихта қалады. Үзіліс математикасы оңай. Әлденіп ал, фантамен рахаттан. Қане, демалатын кез келді, фантамен әлденіп ал. Сабақ арасында фантамен әлденіп ал. Көңіл көтер, қайтала.
Олар жайлы фильм түсірілеміз жоқ па? Кітаптардағы немесе комикстердегі қаһармандар қатарынан табылама. Оның супер костюмын біреу армандай ма? Оның деген сөздері қанатты сөзге айналама. Оларға бұның еш қайсысы маңызды емес.
Мынау сізге.
Оларға әлемді сәл де болса жақсарту маңызды. Міне, осындай адамдарға жусан керек. Жусан, қосымша сен үшін, мүмкіндіктер сен үшін.
Сделай перерыв между занятиями с Фанта. Проведи эксперимент и развлекайся весь перерыв. Вот формула веселья. Фанта плюс перекусы. Ваши игры с друзьями войдут в историю. Математика перерыва проста. Получай удовольствие от Фанта и перекусов. Давай, уже пора передохнуть. Перекуси с Фанта. Перекуси с Фанта между занятиями. Повеселись, повтори.
Им не важно, снимут про них фильм или нет. Станут ли они героями книг или хотя бы комиксов? Мечтает ли кто-то про его суперкостюм? И с какой вероятностью его станет крылатой фразой? Им не важно.
Это вам.
Им важно делать мир немного лучше. Вот для кого нужен Жусан. Жусан. Приложение для тебя. Возможности для тебя.
S.A.S Kazakh Technical University, the general partner of this event, is hosting this year's festival. For 65 years of its activity, KATU has trained more than 70,000 specialists, successfully working in all major sectors of the agro-technical sphere. Among the ranks of our graduates are deputies of the Parliament of the Republic of Kazakhstan, heads of leading enterprises, nationally known public figures, and athletes. Today, our university is one of the leaders of the national higher education on the national rating of the demand for higher education institutions of Kazakhstan, from the IAAR on the quality of educational programs. And also, KATU is the base university for digitalization of agricultural production in Kazakhstan. Our partners include more than 160 world-renowned leading research universities and research centers. Among them are the University of California at Davis, which is the second highest institution in the world of the agricultural industry, Agro-Polytech University in Europe, and many others. Today, KATU has started a dual diploma education in 3 specialties jointly with the University of Milan in Italy, which gives our graduates the opportunity to receive diplomas from 2 countries. Our university offers the opportunity to study at bachelor's, master's, and doctorates. Currently, the number of students exceeds 12,000, of whom 70% are studying under the state grants. There is a full opportunity to study in the military department. All necessary conditions are created for them to be qualified specialists. The university aims for a seamless experience in any format of education. For students, there is a printing office and a library equipped with an electronic system that has more than 1.5 million book collections.
One of the large multidisciplinary education institutions of Kazakhstan with a deep history is S.A.S University. That is, here students and undergraduates acquire practical working skills. In other words, this is a mini copy of a large production and dairy plant. Within the framework of the program of scholarship, academic mobility and double degree education, the university cooperates with more than 170 foreign universities. 150 students a year study for the academic mobility at leading universities in the world. The digitalization process has affected all spheres of society, including education. At S.A.S University, innovations are intensively introduced into the education system, and 3D, VR, STEM technologies are widely used. The IT Center provides consulting to teachers of schools in Shymkent on robotics, provides material resources to students in the Laboratory of Robotics and Mechatronics at the Laboratory of Mathematical and 3D Modeling, and provides material resources of the IT Center to students and teachers of the S.A.S University. In Student Design Bureau, students and teachers prepare scientific projects for participation in regional, national and international competitions. Students of clubs and Student Design Bureau develop robotic systems for participation in competitions and competitions in robotics. The material and technical base of the university consists of 10 educational buildings of a modern standard, 6 student dormitories and 2 sports complexes. For the comprehensive development of students, more than 30 public, political and cultural organizations function. S.A.S University, that has united history and modernity today, prepares more than 27,000 students and invites to take your best.
Сабақ арасында фантамен үзіліс жаса. Эксперимент жаса да үзіліс бойы көңіл көтер. Шаттық формуласы Фанта плюс әлдену. Достарыңмен ойнаған сәттерің тарихта қалады. Үзіліс математикасы оңай. Әлденіп ал, фантамен рахаттан. Қане, демалатын кез келді. Фантамен әлденіп ал. Сабақ арасында фантамен әлденіп ал. Көңіл көтер. Қайтала.
Олар жайлы фильм түсірілеміз жоқ па? Кітаптардағы немесе комикстердегі қаһармандар қатарынан табыла ма? Оның супер костюмын біреу армандай ма? Оның деген сөздері қанатты сөзге айнала ма? Оларға бұның ешқайсысы маңызды емес.
Мынау сізге.
Оларға әлемді сәл де болса жақсарту маңызды. Міне, осындай адамдарға жусан керек. Жусан қосымша сен үшін, мүмкіндіктер сен үшін.
Сделай перерыв между занятиями с Фанта. Проведи эксперимент и развлекайся весь перерыв. Вот формула веселья. Фанта плюс перекусы. Ваши игры с друзьями войдут в историю. Математика перерыва проста. Получай удовольствие от Фанта и перекусов. Давай, уже пора передохнуть. Перекуси с Фанта. Перекуси с Фанта между занятиями. Повеселись, повтори.
Им не важно, снимут про них фильм или нет. Станут ли они героями книг или хотя бы комиксов? Мечтает ли кто-то про его суперкостюм и с какой вероятностью его станет крылатой фразой? Им не важно.
Это вам.
Им важно делать мир немного лучше. Вот для кого нужен Жусан. Жусан. Приложение для тебя. Возможности для тебя.
The Cofounder Technical University, the general partner of this event, is hosting this year's festival. For 65 years of its activity, CATU has trained more than 70,000 specialists, successfully working in all major sectors of the agrotechnical sphere. Among the ranks of our graduates are deputies of the Parliament of the Republic of Kazakhstan, heads of leading enterprises, nationally known public figures, and athletes. Today, our university is one of the leaders of the national higher education on the national rating of the demand for higher education institutions of Kazakhstan, from the IAAR on the quality of educational programs. And also, CATU is the base university for digitalization of agricultural production in Kazakhstan. Our partners include more than 160 world-renowned leading research universities and research centers. Among them are the University of California at Davis, which is the second highest institution in the world of the agricultural industry, Agroparitech University in Europe, and many others. Today, CATU has started a dual diploma education in 3 specialties jointly with the University of Milan in Italy, which gives our graduates the opportunity to receive diplomas from 2 countries. Our university offers the opportunity to study at bachelor's, master's, and doctorates. Currently, the number of students exceeds 12,000, of whom 70% are studying under the state grants. There is a full opportunity to study in the military department. all necessary conditions are created for them to be qualified specialists. The university aims for a seamless experience in any format of education. For students, there is a printing office and a library equipped with an
And we're live. Thank you everyone who continues to watch our final day of the 5th Nobel Fest Lecture Series. It is my pleasure to announce our next speaker, Dr. Pascal Meyer, who is the laureate of the 2022 Breakthrough Prize in Life Sciences, co-founder and CEO of Alphanosos, and owner of Pascal Meyer Consulting. Professor, you have the floor.
Thank you very much, and thank you for inviting me to this presentation. I'm very honored to be here today, and sorry not being here in live in Kazakhstan. I would have loved to do so. So I want to give a presentation that is about one of the concepts and the philosophy which is driving me and which can be seen as simplicity. So I'm unsure whether, yeah, I see it. Okay, great. So that's just a small thing I need to do here is to put this down. So, okay. So I will present this how simplicity can be a paradigm a mental paradigm to turn what was once thought as not possible, unthinkable, into a reality. I will rely on my own experience on this, on two topics, which are next generation sequencing, for which I, as you said, I got the Breakthrough Prize, and next generation therapeutics, as I call them. So to understand a little bit how this was, when we go back for the DNA sequencing world, when we go back in 1996, this was some sort of prehistory of sequencing as it is today. And there was what was called the first human genome sequencing project. It was done by traditional sequencing. That means that you could get 1 to 10 base per second of information. The cost was estimated at $1 per base that has been sequenced. And the human genomes project then took about 10 years and was estimated to have costed about $3 billion. So how was this made? That I cannot have time and to go into all the details. Those who are working in biology or biology students might recognize the Sanger sequencing technology, which was important thing. Two important things is that it was done one DNA sequence at a time. So you needed basically one test tube where you put one piece of DNA and you sequence that piece of DNA. The instruments that were existing were called automated sequencers, and there were these kind of big machines that you see to the right, costing, I don't remember, 50, 100,000 or more. I think it was more, $300,000 or so. What people were doing in order to get the throughput required for sequencing, they were just accumulating many such big machines, loaded, very high tech with lasers inside to make the readings, etc. That was really high tech and massive high tech, or at least gigantus technology. There were also emerging technologies coming out, and here was a picture of what was called DNA arrays that were radio labeled in order to make the readout. And it was thought to use them for sequencing by hybridization. But all these technologies had in common one thing, is that you needed really one test tube per sequence you want to read. And so that means if you want to read 1 million or 10 million or 100 million pieces of DNA, you needed 100 millions of test tubes, which was really close, mostly unpractical. So where this then comes into place where you want to introduce simplicity. The driver behind this is also that there was no choice. For me, for instance, it was not possible to mobilize hundreds of millions of dollars in order to get novel sequencing technology. And so you start thinking of different things. And one of the points that I wanted to put into evidence of what is important when you want to innovate in this kind of things is the multidisciplinarity. Sorry, I'm French, so you need to forgive my charming French accent and difficulty with some words. So what's the background of a multidisciplinary background? So it's basically, it starts very early. It starts basically in high school, and I remember I was using this programmable calculator, which by in the early 80s was something that was already very special, very sophisticated, and I tried to put a neural network on this calculator, which was totally useless. And that also was to say, as you are certainly reading it, I was from a place called Forbach with a singer which may be famous or not in Kazakhstan and elsewhere, which is called S.A.S, but I never met her. So then I went into biophysics studies, so that's already a multidisciplinary field. I learned some maths. I was working on some sophisticated fluorescence work, NMR, and then I developed an instrument, wrote software. So you see, always combining software, instrumentation, optics, and the biology which was behind. When I went to Canada still working on DNA sequencing methods, being in that field, but trying to, not being satisfied with what's going on and trying to find something better. So I went to work on a technology called ELFs, but didn't make big impact, but still that was always that unsatisfaction with what was existing. Then I went also to postdoctoral post in France, in a research institute which is famous in some fields, where I was working on single DNA, for instance, video microscopy, and I was also working on self-structured arrays that could be obtained by not a microlithographic method, but really by physical forces being such that you form micro-sized arrays of structures that could be of some interest. So it's not to say that I have a very good background here. It was a perfect background for not finding a job by that time. But in the end, I eventually find one. And I was, but just before this, I was still, when you have time, what you do when you are a scientist, you sit down and think, basically. And having in mind this kind of array ways of sequencing, very innovative ways that were in the air by that time, but still, for doing such arrays, you needed a robotic system that was taking one DNA piece from one tube, putting it on the array, take another DNA tube from another tube, putting in another place on the array. So it was something that was really cumbersome, even if it's with a robotic system, and not so efficient. So how could you eventually improve on this? And so you start building on your experience. And basically I had that experience with magnetically structured arrays of ferrofluid emulsions. That means that I don't have time to go into the details, but when you put this into a magnetic field between two plates, these emulsions, they aggregate in some sense, and they form pillars that have a certain spacing, which of micrometer scale. So this is some kind of microarray that you can obtain not without having to make them one by one. And this kind of arrays, and these spots make you think also of bacterial colonies as being a biophysicist. That means you know about biology, you know about physics, and you think there's some analogy between here and there. If you can get this kind of things, you can have some bacterial colonies, and then you think bacterial colonies can bring to multiplication of something, and multiplication of something is PCR. I think we are now all familiar with PCR because of the COVID tests, and here is a schematic of how it works. I won't go into time again. The biology student will recognize what's going on, where you amplify a piece of DNA with thermocycling. So you can start from one or a few strands and get up to billions of copies of the same of DNA fragments which all have the same sequence. Then there are also macromolecular biophysics, and when you are working on this, you don't see just the DNA as from its genetic point of view or from its chemical point of view with the phosphates, with the nucleotides, etc. But you see them also as something that is flexible, that has some characteristic scales. And well, what can you do with this? You think, at some point you come to an idea. And as was introduced, that's why I got the Breakthrough Prize through. But I take this occasion at this moment of a speech to give recognition to all the team and all the people who worked with me on working on that technology. It was incepted by me, but then many people were involved at different places, especially Laurent Farinelli, who was with me at the very early times where we started working on developing what is now next generation sequencing, and is the main technology all over the world. So there are many people, as you see, and I want to thank them all. And you can see, if somebody has the curiosity, I have this story on my LinkedIn profile to give a little tribute to all of them. So when we start presenting this, it was already in '98. It was the first public presentation, but the patents were filed April 1st, '97. And in the history of who did what, when in science, it has some importance. And I come to this later because it also will illustrate some of the aspects of what happens when you are innovating, and especially when you're innovating with simplicity. So how does this sequencing work? First thing to do is to form arrays of DNA molecules to have many copies of each sequence, and this works with what was called solid phase PCR and is now called bridge amplification. So that basically you have DNA molecules in solution. They are constructed in such a way that they correspond to other pieces of DNA, the red and green one that are attached to glass plate, and you can then attach the initial copies that you have in one single test tube. You can have many different DNA sequences, and they may eventually attach simultaneously all to that surface. Here I represent only 2 of them, but you can imagine that this is happening simultaneously for thousands of different DNA molecules. Then you take the biomolecular engineering methods. That here it's a DNA polymerase, so it's a protein that makes a copy of a DNA strand, like you would make a photocopy of a paper, basically. And you end up having two strains now attached at the same place. In that situation, you would just separate the strains, wash away, so you have the copies that are now covalently attached to the surface. And now there are two things to take in mind, and they are linked to the macromolecular properties of the DNA, as I mentioned before. First is that you can control the average distance between the attached strains by playing on the concentration of these initial strains in solution. And so you can have them more or less spaced at will. The second one is that because of the physics of the DNA molecule, when it folds back and eventually finds another primer on the surface to where it attaches again, it's at a distance that will be of the order of a few hundred nanometers. This is something you know. Even the DNA molecules can be much longer, a few micrometers in size. The end-to-end distance that you would get is a few hundred nanometers. And so when this happens, you get, again with polymerase, a novel copy which is also attached to the surface, but which is as a distance which is much smaller on average than the distance between the two initially attached strains. And so when you repeat this, you get what is called DNA amplification in the jargon of molecular biologists, and you get several copies that are all attached to the same point. When you look them on the top, you would see something like this. Here, each color represents a different sequence in what you obtain, and I called these DNA colonies because, by analogy, by the way, bacteria multiply on agar plate, and here the DNA amplification is more or less has a lot of analogies with this. So this was initially coined DNA colonies, and to date has been remained, and I'll come back to this renaming matter later. They have been renamed as DNA clusters. So each DNA colony contains potentially a different DNA sequence, and all this comes from one single test tube. So you don't have to have 1,000 different test tubes and take one, attach it to the surface, take another one, attach it first. It's very simple here. You have all the molecules in one tube, you spread them over that surface that has the primers, and you get an array with all the different sequences. So the next thing, which is very important also, and that's one of the key factors of the success of that technology, is that it has been designed in such a way that you do not just amplify a single DNA sequence. When you're making the COVID tests, basically you have, I could have written COVID here, for instance, but you are going to target one piece of DNA in the genome or wherever else. And you have what are called primers. They are called forward primers and reverse primers in the jargon of microbiologists or of molecular biologists. And they are specific to that piece of sequence you want to amplify. And so you can make many copies of that piece of sequence, but only of that one, which is one of the great things about PCR, and for which there has been a Nobel Prize in the 80s. And so now the answer you can get here is to say, okay, I was looking whether Paul was in the room, and if I get an amplification, I know that Paul was in the room. Or says, oh, that means that that sequence was on my sample. But you need to interrogate one after the other. So if you want to make to look at several of them, you need to have several test tubes, each with a different interrogation. So one of the things that has been brought forward with the DNA colony sequencing, the next generation, is to be able to put together in one single test tube that you can interrogate many different sequences. And it goes the following way. That's you start cutting the DNA into pieces, and there are different ways of doing this for molecular biology. They have molecular tools to do it. You could just fractionate it by mechanical means, and then you can tail them with is now called universal primers. Here you had primers that were specific only to one piece of sequence. Here you have just one pair of primers, and you tail them in one test tube to all the fragments that you have got, and you got them also in one test tube. And so you form the DNA colonies as you had before, and now you have different colonies having each corresponding to a different primer. So you see the big difference of simplification here was to think of something that initially would have taken for millions different sequences, 1 million test tubes, to something that for 1 million or 10 million or more DNA fragments, you had only to work with one single test tube. So we then turned this into experiments, and they were successful, and they have been presented also in '98 at the first conference. And basically we showed with other data, explain how this was made, that we were obtaining DNA colonies that could be visualized by fluorescence, that the average distance between them was of the order of few micrometers, and the size was of the order of 1 to 2 micrometers, and also showing that, as expected, the number of spots, the density of spots can be controlled by the concentration of these tailed fragments that I described earlier. And from that, you can already deduce that you can get 1 to 10 million colonies per square centimeter. And if each fragment has a size of about 1,000 nucleotides, that means that you can sample a whole human genome in pieces of 1,000 nucleotides on one single microscope slide. So remember, before the image I showed you had hundreds of instruments taking a whole factory of sequences, and here on one single microscope slide you could eventually get a representation of the whole human genome in a way that you can read potentially the sequence. So the sequencing results were shown also. So we showed the principle of it. These are images that are really from that '98 conference. The only difference being there was a blue background, which was very fashionable by that time, and now I turned it into a white background, which is better looking, I believe. So basically, we described how we were making the sequencing, which was stepwise sequencing. Later, we found out that this was not new, but maybe it was the first time that it was reduced to practice with fluorescent detection, however. So basically, you make what's called base by base sequencing, or sequencing by synthesis. So you interrogate, does this nucleotide, I think all of you know that DNA is made of 4 types of nucleotide, A, T, C, G. And so you integrate, does this A nucleotide is part of a sequence, yes or no? Then you integrate, is the C part of that sequence, yes or no, etc. And you do this sequentially and repeatedly, and you measure on each colony, you may measure the signal that you get. And if the nucleotide is incorporated, you get an increasive signal. If not, the signal stays constant, etc. And so in '98, we presented the first experimental results on this, showing that we could see the colonies that were revealed by the incorporation of the first nucleotide. Then we put a second one, and we saw that some colonies were appearing while others were getting of higher intensity, and that's what we were expecting, as these are the 2 fragments that were in that sample. That one was incorporating a C and a T, and the other one only a T, and that's corresponding to what we get. We had only these 2 nucleotides by that time because they were the only one that were commercially available, and so we could not make the experiments with additional nucleotides by that time. So the objective for us was to make 10 base sequencing and eventually 20 base sequencing. And like some expert may debate of this. I take this debate later on if you want. So indeed, that with that level of sequencing, you can already get interesting sequencing results. Of course, if you can read more, the better, but with that minimum, you can already do a lot of things. And when I presented, and here's something very interesting. I had to make a lie, actually, because when you take all these things together and you make the calculation, you come to a conclusion that you would be sequencing a human genome within 1 day for a cost of a few $100s. That this was something my supervisors and told me that, okay, you should not say it. So we said, okay, we kept it very low, that we were sequencing only at a few tens of bases per sequence, 10 to 100 times more than the classical, the standard by that time. And that the costs were also, okay, we were $1 for 6,000 to 60,000 bases. But this was a fake announcement because we knew that already we would be sequencing at much higher rate once the technology was fully mastered. So now just a few words about other things that you face when you are developing novel technologies, especially when they are very simple. The people later may eventually rewrite history, and that has been done until the breakthrough prize by let's say the USA, that they really were writing that the invention of next generation sequencing was due to the company Solexa in UK, and the two founders got the breakthrough prize with me. It's deserved. I have no problem with this. But just to say how the story was then retranscribed by people who were writing books about this, and first they said that what Solexa was doing from the people who were working in Solexa was saying that didn't work, and they bought the technology that we were developing in Switzerland about this. And once they bought it, suddenly they could have a working technology. But they never cited it, neither myself nor anyone, or no the patents, nothing. That is just the word appears a few times, and that's all. It was the name, Mantea was the name of the company we were working on. So basically, I wanted to give some visibility on what I call the Lebanese branch of next generation sequencing, which started when I arrived at GlaxoSmithKline Geneva Biomedical Research Center, or Institute, I should have written Institute, and which was then took over by a company named Serono, and which renamed it, where we made this first proof of concepts, and then we founded a company named Mantea Predictive Medicine in 2000, basically started working in 2001, where we wanted to develop that next generation sequencing technology in order to lay the basis for personalized predictive medicine, and we were thinking of making a genome card. So where we were in 2003, where we had functional sequencing instruments in place, and here again, you see how you can make things with simplicity, basically. When you realize that you need to make a microscopic fluorescence observation, instead of building a whole new instrument, you take a fluorescent microscope. You need an XY stage, which was assembled on purpose. You had pumps that you could just buy, you had filters, you had a computer, and basically you needed also microfluidic channels. Actually, this was interesting by that time because the right size of what you wanted to get was really to have something that's 50 micrometer deep channels, because you don't want them to clog. And by that time, microfluidics and microlithography were very good in making nano-sized structures, or 1 to 2 micrometer sized structures, or etching channels, but channels that were 1 to 2, 3 micrometers deep, not more, which would clog very quickly and not necessarily practical. And so we found a way, which I worked earlier on while I was in one of the postdocs, that was to have a plastic structure that was 50 micrometers thick, in which the channels were present, and they were between two glass slides. And this was what we had by 2003, and it's something that is still, from what I know, used today in the next generation sequencing from Illumina. So we had to develop also some autofocus based on just image variants, and by that time it was not available. I think today it's quite standard, but by that way we had to write it, the software ourselves and the algorithms ourselves, and autofocus was eventually made using lasers, using complicated things, and again trying to make simple things. You get this kind of images of DNA colonies that here are detected by fluorescence. And one thing that you need to do is to quantify the amount of light that comes from each of these spots. And this can be very complicated. It's not that simple that you just have a balloon that you can remove. It's more complex. And so we had to develop algorithms that are able to make this with cellular automata that were giving then files that had just the position of one of the spots and the light intensity in it. And from this, we could deduce the sequence, the first sequencing reads we were making. This is one of them. Another thing that is important was always to overlay the two images exactly, because the mechanical XY stage has a precision of a couple of very few micrometers, but it's too much looking at structures that are spaced by a few micrometers. So you cannot guarantee that each image overlays exactly. So there was a software treatment in order to allow this to be done, rather than trying to get mechanics with a precision of less than a micrometer, which would have been very difficult, especially by that time. So this kind of results we were getting, a piece of sequence that was sequenced, so which we integrate, does T belong to the sequence? No. C, no. G, yes. A, no. T, C, no. G, yes. And you see them here on the images. Here there's a tiny red cross. And no, no, yes. No, no. The G, yes. No, this is here, the G, yes, etc. And you can read that way. You can read the sequence within the DNA strand that is within these DNA colonies. So by that time we were at 9 nucleotides, 10, but then something happened. That's always part of when you develop technologies, and you need also to be in the right place, really. And that's not novel, but something that's very, very true. So in summer 2003, the mother company who was financing us said they would stop financing it, and they sold, as was written in that book from Davis, that it was sold to Solexa, and then later Solexa sold it to Illumina. So this is, in a nutshell, the story of next generation sequencing, seen from one up to the breakthrough prize forgotten part of it, which was the work that was done on the Geneva Lake region. So another example of how to put simplicity into work is what I'm doing actually right now, and it raises again from questioning. And you, of course, at some point you need to have some time to think about this, but what is odd with traditional drug development? And when you think about it, genetic analysis points to multiple targets. That means that one of the driver of sequencing the genome was eventually to find the genetic roots of disease. And very quickly, people start realizing, thanks to next generation sequencing, that there are many genes involved in diseases, not just one gene that is involved on which you may eventually act. Diseases are multifactorial, so it's not only the genome, but also the environment. The bacterial biofilms and bacteria might be involved in chronic diseases. Most drug development fails because of undetected toxicities. So I think people work 5 years on make experiments on mice, etc., phase 1, but still you're not able to detect accurately, with confidence, the toxicity of novel drugs. Precision medicine is not a guarantee for safety or efficacy, as we will see also. Single compounds acting on a single target, that's an utopia, and it's accepted now. Drug development, as everybody knows, is long and costly, and chemical synthesis is not sustainable and is disastrous to the environment. So you think I'm exaggerating, and also the availability and to everybody is an issue. So if you think I'm exaggerating, I put some examples. Here is a good example of precision medicine. So one gene was identified, an antibody acting on that gene specifically was developed. It was not toxic in mice or in rodents or in whatever they has been tested, and they put it on the market. They had some good early clinical results, but still there were some people who died of a drug and they stopped producing it and selling it. So that was close to $1 billion going down the drain. Precision medicine. So an example of multifactorial. So this is a result of genome-wide analysis where it reveals that there are many genes involved in that disease. The implication of bacteria in disease that has been long ignored and is coming very slowly back. So this is a publication of a group of a Mayo Clinic in the USA. So enough, I think, very prestigious. And they were seeing that bacteria associated with breast cancer. So they saw that in breast cancer biopsies, in breast cancer biopsies, they found bacteria which are different from biopsies from non-cancer breast biopsies. And so two things. First, there is a difference, and also there are bacteria present even in a healthy person. So we are very likely infested by bacteria everywhere, and there has certainly roles in many diseases. Pollution, it's now well documented that antibiotics make a big pollution into rivers and hinder fish development, and even are problematic to human health, especially by the development of resistance to antibiotics by these bacteria. The pollution of producing drugs is tremendous also, so that for instance, in the European Union, there's nearly no drug produced anymore because just the chemical synthesis is so disastrous and dangerous that they do it elsewhere. And it takes time to develop drugs. So even vaccines, even with the latest technology, they come late. And there are some examples of riots for having access to drugs in the world. So all these things, what can we do? So it's the first step, as in every situation, is to turn to existing science. And there's novel science coming out since some years now, 10 to 20 years, that cellular systems perturbation could be an approach that could bring some novelty and some solutions into drug development. And what this basically means is that we take plenty different of compounds acting simultaneously on many different targets. So that's in concept, yeah, that's nice, but how can we achieve this in a tractable manner? Discovery time, costs, and safety, because you imagine already the difficulty to produce one single chemical compound that is not too toxic. Now you have plenty of them. It would be a nightmare. So what could be the solution is that think of how to make this much simpler. That is that rather than trying to make a novel compound safe, why not looking what you can do with compounds that you know that are already safe? And we know where to find such compounds. They are in edibles, and in edibles there are hundreds to thousands of compounds, chemical compounds, that we can ingest, that act on our organism and that are not toxic. So can we have efficacy? Well, extracts from single plants are known to have some efficacy, but there are a lot of critics to it. So how could we improve on classical plant extracts? So the answer is found in some place. I wrote some theoretical background on this. I will go very quickly through it for a few of you. I was told there were many students looking at it, so maybe there are students that have some mathematical acumen, so they will eventually follow. The others, I go very quickly. It's not too important, as you will see. It's basically what is described is that you take a large diversity of chemical compounds. These compounds will act more or less on each different protein or target in the organism. And so in that sense, you can have perturbation of this protein. This can be just because they change a little bit the pH, or that they interact with some amino acids in the protein. They make some allosteric modulation or pseudo-allosteric modulation, etc. So that's not science fiction, that's known. There are many arguments on this. If you do this, you would perturbate cellular subsystems. So that means you can already perturbate bacteria or cellular organelles. If you do this, you certainly have an effect on the organs themselves and in the end on health. So each of these elements can be called an endogenous biological feature, and the metabolites are either endogenous features or nutrition's exogenous features. And what you can then do is to write something very simple, that is that the amount or the magnitude, or however the measurement you can do on one of these biological features can be written as some homeopathic value plus the perturbation that is brought by a particular chemical diversity to which you submit the organism. When you do this, that can be rewritten, and you can see that this is in the end very simple in first approximation, is that the effect that you get is just a simple calculation from all the nutrients that you would submit to the organism. And what you can show is that the effect you can get can be eventually as strong as what you could get with one single compound like a drug. So when we look at the distribution of the deviation from homeostasis, which is maybe what you're interested in, and of course, when you make such a sum, and those who have some mathematical skill will understand it, is that on average you get a zero effect, because some effects are positive, some are negative. If you have plenty of them, the average is zero, but there's some distribution, of course, with some standard deviation. And it can be shown that this standard deviation is decreasing with the complexity of the organism and the chemical diversity in nutrients. So, and this has very important effects. If you see what I wrote on the left is the blue curve, and if you have only a standard deviation that's 10 times smaller, you see that there are plenty of parts where the probability of having a strong effect exists for the small deviation, but is non-existent when you have a smaller standard deviation. And even if you have another factor of 3, you go to something that has still a smaller standard deviation. What this means, and then you come to something much simpler, is that you can have a probability of having strong effects on less complex organisms, but with mixes that won't have a strong effect on organisms of high complexity. So when we translate this into simple words, is that basically you can have cellular system perturbation with massively diverse mixes of compounds, and that's what we are doing when we are eating. We are exposing our organism to massive perturbation with many different chemicals. Most of them are of no effect, but some of them you could find a way to have them having a very important effect. So how to do this in practice? Basically, you think of edibles. When you're making water extracts of mixes of edibles, in other words, you're making alimentary preparations. And when you're doing this, in fact, you are making massively parallel phytopharmacology, but it's naturally safe. You don't worry about the safety of it because we do it all the time. And an important point, if you can show that the mixes that you assemble are synergistic, you can patent them. But the issue is that from all the possibilities, traditional brute force would not be an option because there are too many possibilities, 10 to the 10 to 10 to the 40. And so you need to find smarter way. And to do it, AI would be a possibility to do it. So this has been applied, and give as an example the development of such an edible preparation that has an anti-coronavirus effect. The way we did it, we worked with vero-EC cells infected by the coronavirus, and normally these cells die out if there is no treatment. So then we assembled mixes of water extracts of edible plants, and we made the experiment, and here each bar represents the survival in one experiment. So the first mixes are assembled at random, and we see that there is very little effect, if any. But this data is entered into the algorithms, and these algorithms then learn the relationship between the composition of the mix and the eventual biological effect that you are measuring. And so by doing this, once you have this, they suggest novel mixes to be tested, and they are tested. Here are the light blue bars, and there's already some improvement. A second iteration, we get the dark blue bars, a third and a fourth in red blue bars, where we were able to get nearly 100% survival of the cells despite the infection by the coronavirus. So we stopped here very early on, and we went into a mouse model. So there's a lot of critics against this mouse model because all the mice die. The only treatment that has some effect was the administration of convalescent patient serum, so antibody therapy intravenous. And then we can discuss whether it's a good model or not. It's a debate we won't have here. And here are the results that we get. So we were following the survival, the morbidity and the body weight of the mice that had been infected with the coronavirus, and a group was treated, another group was not treated. And it was the group that was not treated died out after 9 days. All the mice were dead, whereas the treated ones, we had 30% survival, as good as with the serum of recombinant patient, knowing that here the administration of the water extract of the edible plants was done by ingestion per roast. So they were eating it, but still it kept the effect we were seeing in vitro. And so these are real survivors, as could be seen when you follow the body weight, especially of the survivors, that you see that the survivors, they recover their body weight. They are real survivors, even if the experiment was stopped, the follow-up was stopped after 10 days. So here that means that we were able to develop an anti-coronavirus within within 2 months, basically around Christmas and New Year, being in end of 2020, while there was restriction on everything, as you all know. Basically, it was 5 iterations. So these results could have been reduced to 2 weeks because it takes 3 days to get an experimental result, and the next day you can do another experiment. So it can be very fast. So where we stand now is to eventually enter this into a clinical trial, because what we have is recognized as being an edible. It's a food supplement by composition, and it's indeed possible to have this product as a food supplement, as long as you, of course, you don't claim it's an anti-coronavirus. It's good for your health. That's all you can say, basically, but still, that means that by composition, it's something that can be produced, administrated without any problem. So this brings me to the end, nearly to the end, to what I wanted to show, just to say what we did with the coronavirus can be done on bacteria, can be done on cancer cells. What has been done on bacteria have some clinical benefits, as shown by clinical by trials on patients. We have efficacy also on bacteria that are of high concern for gonorrhea. And these extracts, they work for bacterial infection in mice, as we have also shown in a mouse model. So I think I talked already a lot. So I come to a little bit to my conclusions. Everybody who would have a question can, I'm very happy to answer questions by mail, or if you want to contact me. So I wanted to make what I can do with these novel ethical and with these novel edible treatments. Basically, that's a hypothesis-free approach, so you don't have to make hypotheses on the molecular level. You make just experiments, and the experiments eventually, if you have making the right experiments, you're developing something that has the expected effect. It's a production that is fully sustainable without industrial hazard, because it's basically an alimentary preparation. So with water, no explosive or inflammable solvents. The treatments become affordable. That's very important, because we have seen it with COVID, but it's not always possible to have treatments that you can deploy all over the world because of costs, because of production issues. And even here, with this kind of approach, you could adapt each treatment to the local botanical resources of the country to which you want to bring it. For instance, to adapt it to botanicals growing in Kazakhstan, or botanical growing in Uganda, or wherever you want. It can still be patentable, which is important to finance clinical demonstration and later commercialization, because people who distribute these and produce these therapeutics, eventually they need to make a living too. So patent is very criticized, but it's important to get products to the patient in the end. The composition is fully biodegradable, so it's very important for antibiotics, because traditional antibiotics that pollute rivers and you have problems, whereas with this, these are botanicals and no botanical ever accumulated in nature, so they are degraded. That's always possible. It's very rapid as a discovery, as I told, so it can be discovered really within days if you master the experiments to be done. So think fast-growing bacteria, you could make an experiment every 3 to 4 hours. So within a day and a half, you may eventually develop a novel extract that is acting against such a bacteria. So this opens the way for rapid response to pandemics, especially because what you get is by composition an edible. As long as you don't claim that it has a pharmaceutical effect, it's an edible. And so you need to take a decision. Do I let people die, or do I give them something that's an edible and that may eventually be helpful to them? I don't have the answer. I'm not a doctor, nor a politician. The other thing is responding to the emerging antimicrobial resistance crisis. And there's a lot that is told about it, but there is no solution. Here we have a solution, potentially. The resistance is not developing as we could test, or you would anyhow expect it to develop very slowly. And as we have already early results on cancer cell lines, so far in vitro only, but still that may open a new possibility for cancer treatments. So the question that comes always in this kind of cases is this too simple to be true? And so I come to my conclusion, basically to wrap up on the lessons that I learned. I have learned many of them the hard way. So if for innovating and or using simplicity, basically you need to praise transdisciplinary education, because very rarely do you have in one specialty a solution to a problem. You need to put different things together, and in order to be able to do this, you better to have a generalist education. You need also to practice this transdisciplinary thinking, because that means that you need to be willing to solve something and to use, say, what can I do if I take things from different areas and put them together. Important teamwork. Once you have an idea, to put it into practice, you usually cannot do it alone. You need many people adhering to the project and make it happen. The other thing that can be a little bit disappointing is that when you think of something new by putting many different things together, sometimes you think some of the details are novel and in the end they are not. So it's something you need to be conscious of and recognizing it, that other people already invented a part of what you are using, where they didn't have a full picture. The big thing is facing incredibility and indifference despite the scientific evidence. And I had this a lot. So the first results I show you on the conference of '98, I had, well, there was 1 or 2 persons that eventually seemed to be interested, but nobody was really paying attention to it. And also, I had also people making fun of me because I was using a microscope to make sequencing. You know, the people were used to have these big machines with lasers, with capillaries, with complicated things. And you say, okay, I take a microscope, take a glass plate, I make some just PCR, and I would be able to sequence that. No, it cannot be true. Corporate and national politics, I saw this all over. You saw it for the sequencing, where the mother company decided not to pursue that project. And for today, we have some, I believe, somehow interesting results for a potential anti-coronavirus treatment, which waits just to undergo clinical trial, and nobody seems to be interested in it. So what can I say? Facing a lack of recognition, it took, as you saw in that book of Davis, that none of the people who were working on next generation sequencing on the Geneva Lake on my side were cited in anywhere. It was just blacked out. It was a company who had the technology. Okay, I'm very grateful to the Breakthrough Prize Committee to have to have put this into the light again, and that now people know that there was a group on the Geneva Lake with myself and other people around, especially Laurent Farinelli, who were working on this and were key in getting this happened. And in the end, what you need to be perseverant and keep marching, as I wrote, despite contrary winds. In the end, eventually, you were right, and was predicted, was expected by theory, by many things. It takes a long time until people are ready to accept what is in the end just scientific evidence. But it's a little bit like black holes, or not exactly, but everybody believed in black holes since a long time, and the first one was only observed a few months ago. So in physics, this is much more possible than in life sciences, but that's another discussion. Okay, so I would like to thank you all for having followed this long and I hope not too boring presentation. And I thank again the organizers and everybody. And I'm, as I said, ready for any interaction through, I guess, email after this.
Yeah, thank you so much. It was a really interesting story with like lots of life lessons, as well as some very useful information on how it's possible and how to approach things. So I think we have just a couple more minutes, so unfortunately we will not be able to take all the questions, and we will definitely share the contact details with those who want to answer answers. So maybe just one. So one of the students is asking, so when we're sequencing samples, sometimes we had not very clean results. How to, maybe some practical tips on how to make them better.
Okay, I'm sorry, I didn't get the beginning of your question.
So when we are sequencing samples, sometimes we have not very basically is, do you have sort of tips on how to improve the situations when the results you're getting is not exactly what you had in mind initially?
I must confess, this is a little bit too technical a question for me today. I haven't been using sequencing since a very long time, and I won't be able to answer it, unfortunately. I'd be of no help.
Okay, definitely no problem. Thank you so much. We really hope to see you one day in person, maybe next year in Kazakhstan. Thank you so much.
Definitively. Thank you again. Bye-bye.